Yes. So we are already 5 minutes late, only, and this is good. So we will try to let's try to stick as much as possible to the time. Well, I'm not sure coming from the CNRS in Montpellier in France. We have consciously left a lot of time for discussion. We have plenty of discussion then later in the afternoon. We will be then trying to summarize all notes in the discussions so that there will be one person in charge of taking notes in the discussion so that then we can share all these thoughts. And so let's try then also for the speakers now and for all. So let's try to stick really to the time. Let's try to have at least some 10 minutes for the discussion for the keynote speakers and some 5 minutes of discussion for the presentations that are shorter. And then we have now all the block of non-genetic inheritance. So I'm extremely happy to say that Carmo-Fonseca accepted to be here. She's, as most of you may know, one of the main names in the world of RNA and RNA biology and all the transfer, the key role of the RNA layer in information transfer. Thank you very much. I'm really happy you're here. And then we will have talk about Mariana Schiavon and Anastasia Bresenko also going with this non-genetic inheritance and how this epigenetic switch that we will see here and all the maternal inheritance effects are going to be. So don't take much more of your time. And again, let's try to stick to the time so that we can focus on the discussions. Please.
Okay, so I want to start by thanking the organizers, in particular Inácio, for this very kind invitation. It's a real privilege for me to be and to speak in this workshop, and I'm very much looking forward to very exciting interactions. So today I will speak about the boundaries between splicing, RNA splicing variability, in disease. And as I was listening to Francesca's talk, I realized that I have this bias of thinking about variability in a negative sense, because for us in a medical environment, variability means errors, and these errors tend to be deleterious. But I think we can discuss more about this at the end. So we're going to talk about RNA splicing, and I would like to start by the evolutionary origin of splicing. So we all know that splicing of introns from pre-messenger RNA is a hallmark of eukaryotes. Yet splicing probably was already present in the very early moments of the emergence of life, because we do find RNA elements with the ability of self-splicing in bacteria and in mitochondria. And these self-splicing elements, they have all the characteristics of mobile genetic elements. So one of the prevailing ideas about the evolutionary origin of splicing is that introns in our pre-messenger RNAs most likely arose from the invasion of these early, of our early eukaryotic ancestors by these genetic elements that had already this ability to self-splice. Okay, but once introns became a part of the protein coding genes of eukaryotes, what happens throughout the emergence and the evolution of more complex organisms, there was a tremendous expansion of the number and the size of introns. So when we look at the more simple organisms like yeast, they tend to have very compact genes with few introns and very small introns. And as we move in complexity, the genes start to expand in size, and they expand mostly due to an enlargement in the size of introns. And in humans, we have extremely long introns. This is the dystrophin gene is the longest human gene, and it is very long because it has very long introns. And as you can see, if we compare the percentage of exon sequence in C. elegans and then in mammals, in mice, and in humans, we see that there was a progressive decrease in the percentage of exons because introns have become gigantic. And what is curious is that although mouse and human introns have similar size, their sequence is different. And it's different because the introns expanded due to the expansion of repeats, and repeats tend to be species specific. Now, what is the molecular machine that is responsible for identifying where are introns and where introns should be removed from the pre-messenger RNA? This molecular machine is called the spliceosome. It's a complex machine composed of small RNAs and many proteins. In total, there are over 170 proteins present in the spliceosome. In this structure, you see how this big complex is able to attach to both ends of an intron, bring the two ends together. Here is represented the intron, and then catalyze the ligation of the two exons. How does this molecular machine recognizes where splicing should occur? It's all about, or at least we think that it's all about recognition of sequences, and we have two levels of sequence recognition. We have conserved sequences that highlight where are the ends of exons in the beginning of introns, and this is critical for the spliceosome to know where it's going to cut and ligate. And then the spliceosome works together with a number of auxiliary splicing factors. They are RNA binding proteins. They bind to their preferred sequences, both on exons and on introns. And there are numerous interactions between these auxiliary factors and the spliceosome, and this crosstalk eventually determines where splicing is going to take place. Now, the spliceosome tends to be highly precise in recognizing splice sites, yet there is variability. And sometimes the spliceosome makes mistakes, or the spliceosomes does the splicing in a slightly different site. And I will start by showing you now our own work on looking at the diversity of splicing choices in a sequence-independent context. So looking at one particular gene that gives rise to a pre-messenger RNA that must be spliced, because this is a long gene, this is a sarcomeric gene. So it's a gene that is present in cardiac muscle cells. It's like the dystrophin, it's a very long gene with many exons and introns, and all of these introns must be precisely removed. Yet, now that the splicing field as a new technology, which is long-read sequencing, we are starting to quantify the splicing decisions in a much more detailed way. We are not just focusing on one particular splicing event. We can look at the entire mRNA and see what were the splice decisions that were taken by the spliceosome when it was processing this transcript. And what is very well established in the splicing field is that not all splice sites are equal in terms of their strength. And when some splice sites are weaker, then it's not surprising that these weaker splice sites tend to be more variably recognized by the spliceosome. Just to give you an example of this connection between weak splice sites and variability of decisions, in this pre-messenger RNA, there are a few microexons. Microexons, as the name indicates, are exons that are extremely tiny. In some cases, in some extreme cases, like microexon 14 of this gene, it's only 3 nucleotides. So it's one exon encoding one amino acid. And as you can imagine, such a tiny exon poses a big problem for the spliceosome to be recognized. And so where microexons are present, there is more variability in splicing decisions. And I try to illustrate this variability here with the results of long-read sequencing that we have been doing in this gene. And although the majority of messenger RNAs are correctly spliced, so they have all the exons properly ligated, there are a few mistakes that I'm highlighting here with these red asterisks. And they are different choices all around these weak problematic microexon. But the vast majority, over 95% of messenger RNA that is present in the cell is correctly spliced. So we tend to say, okay, this variability does not matter. What I want to stress is that when we measure this 95%, we are ignoring a lot of the variability because most of these misspliced mRNAs, they are recognized by a quality control mechanism in the cell called nonsense mediated decay, and they are rapidly degraded. So when we say that the vast majority of messenger RNAs are correctly spliced, this is ignoring that more than 5% of the transcripts were not correctly spliced, but they are not present because they have been degraded. And we can quantify those experimentally. Now, what I would like to focus now is, okay, we have this variability or these errors. When are these errors tolerated and be part of normal physiological variability, or are there specific contexts that are going to make these errors deleterious, or in the extreme, when are these splicing errors always present and leading to a disease? So I will now briefly introduce diseases that are caused by splicing errors. And for many years, the majority of diseases that fall in these categories are diseases that are associated with mutations in those essential splice site sequence motifs. But for me and many others who know about all the complexity of regulation of splicing, we predicted that there are many other mutations out there affecting the other sequences that are important for splicing. And many of these sequences are located deep within the introns, and they tend to be oversighted by the standard genetic medical diagnosis because the standard testing is focused only on exons, with this idea that it is the sequence, the coding sequence, that causes disease. So a few years ago, a PhD student in my lab, Rita, did this careful curation of the literature, looking for cases where mutations located deep inside introns were established as causative of disease. And as you can see, the number was extremely small. So only a few diseases were known to be caused by mutations in introns. And this stimulated our lab to go deeper into this question. And we started the project trying to explore the intronic sequences, the intronic landscape of large genes that are known to be causative of disease, but were not yet extensively studied. So as a model diseases, we choose this inherited cardiac disease. Is called hypertrophic cardiomyopathy. Is the most common genetic heart disease. It's a very important cause of sudden cardiac deaths, namely in athletes. And the most commonly mutated gene is this myosin binding protein C, frequently contains splicing mutations. But the splicing mutations that were known were restricted to the canonical splice site. And approximately half of the patients that have the clinical phenotype, they don't have a causative mutation. And our hypothesis was these patients are underdiagnosed because we have not looked at their introns. So that's what we did. We initiated a collaboration with a group of Professor Perry Elliott at the UCL. They had over 1,000 patients with this disease for which they had sequenced the entire genes. And what we did in our lab, this was the work of another PhD student, what we did was to apply an AI-based algorithm that had been published in the meantime in 2019. And it's now a very widely used prediction tool. It's called Splice AI. And it basically predicts variants that are likely to disturb splicing. And when we did the screening of introns for these 1,000 patients, we did find 4 variants located mostly in intron 13, which is exactly the intron preceding microexon 14. And we decided to focus on one of these variants for which there was a very strong evidence that it was indeed a disease-causing mutation, because it was possible to demonstrate co-segregation in 3 families. Co-segregation means that affected members of the same family, they all have the mutation, whereas non-affected members, they don't have the mutation. What do we use in the lab to experimentally study a mutation? We use iPS-derived cardiomyocytes as models. So we induce pluripotent stem cells from the blood of patients, so we can reprogram white blood cells into these induced pluripotent stem cells. And then we use CRISPR. This is the work of a postdoc in the lab. We use CRISPR to correct the mutation in the patient cells, or the other way around. We use iPS lines from healthy donors, and we introduce the mutation. We knock in the mutation by CRISPR. So like this, we have isogenic iPS-induced pluripotent stem cells. We can then induce to differentiate into cardiomyocytes, and you see this remodeling of stem cells. After 30 days, these stem cells look like these. They are elongated cardiac cells with beautiful sarcomeres. And when we do single cell RNA-seq, the vast majority of cells in our cultures correspond to cardiomyocytes. Now, when we repeat the same sequencing analysis of these gene in the mutant cells, what we see is that basically the same errors that were present in the wild type cells are there, but now they have increased in numbers. So what we see is that now we have more transcripts that have these different types of missplicing. And so the total amount of correctly spliced RNAs has decreased, and the proportion of the misspliced transcripts has increased. So basically, the mutation is not creating anything new. The mutation is just changing the proportion of the errors that were already present in an independent, in a sequence-independent context. Does this matter? And so to see if this change in the relative proportion of normal RNA, misspliced RNA, whether it has a phenotypic consequence, what we do is a number of structural and functional characterization assays of our cardiomyocytes. And what we can observe is that the protein levels, so just to go back, so I showed you here that the total levels of correct messenger RNA is decreased, and as one would expect, the protein levels are also decreased. And as you can appreciate, the structure, the morphology of these cells is dramatically different. The cells become larger. The sarcomeres are much more disorganized. And when we measure the ability of these cells to contract, because these cells are contracting, they are beating like the real heart, we do significant changes. So this particular gene called the myosin binding protein, it's represented here. It's a protein that is involved in the interaction between the myosin and the actin filaments that is crucial for the contraction. And the idea is that when there is not enough C protein, then this sliding of filaments is not occurring in a normal way, and therefore there are contraction problems. Now, coming back to the problem that stimulated our study, we are trying to show that intronic variants could explain this large proportion of patients that don't have a diagnosis, and we could find 2%. So we were happy that indeed there were mutations in the introns, but we were far from explaining the 40%. Something was missing. So one possibility is that these patients have mutations elsewhere, but another possibility is that the Splice AI tool was not predicting all the intronic mutations present in those introns. And we wanted to explore more this possibility. And as any computational tool, any prediction tool, you can play with the thresholds of the tool. And initially, we wanted to be very conservative, so we were focusing on mutations or variants that were predicted to affect splicing with a very high score. But then we said, okay, let's lower the score and see if we get more mutations. And yes, we did find more variants. And in fact, we were very skeptical because we were now using very low prediction scores. But when we reprogrammed IPS and differentiate cardiomyocytes, yes, the tool was correct. Even at low score, we could confirm that there was a splicing defect there. So this made us reanalyze what threshold scores should we use to apply this Splice AI tool. And what Pedro did was to go on and update the list of curated mutations that Rita had done in 2017. And in 2023, obviously, there were more intronic mutations reported in the literature as causing disease. And Pedro used these now ground truth mutations as a benchmark to see how the Splice AI tool performed. In the meantime, there is another tool available called for splicing prediction called Pangolin. And what he realized was that both Splice AI and Pangolin had very high specificity and sensitivity identifying these ground truth pathogenic mutations using a score that was quite low. This was unexpected, but made us trust that we could use this tool at such a low threshold. And then a more recent student in the lab, because in the meantime, Pedro graduated and moved away. Now, Pedro Silvestre has been using the Splice AI tool with these low scores to look at variants that are predicted to affect splicing in the normal human population, in the general human population. And for this, we have been exploring the GNOMAD database, which spans thousands of whole genome sequences from individuals of diverse ancestries. There are millions of variants described. And what we asked for, of all the variants in the human population, how many are predicted to affect splicing? And the result is, I don't know what you would expect, but the result is that the proportion is extremely small. So if we use the low splice AI score, we have the most 1% of all variants in the human population are predicted to affect splicing. And if we go up with the score, the number of variants decreases and it can go down to 0.02%. The majority of these splice disruptive variants are very rare in the population with very low allelic frequency, suggesting that they are purified against. So it's not good to have variants that may disrupt splicing. And the prevalence of these splicing disruptive variants is generally proportional to the total number of variants observed in the gene. However, there are differences. There are groups of genes that have much less splicing variants than expected from the total number of variants. And curiously, the DMD, the dystrophin gene, is among these. So suggesting that it's not well tolerated, even when there are long introns that would be prone to many genetic variation. It's not well tolerated to have variations that would affect splicing. And now that we have this perspective from the general human population, we wanted to go deep into the disease context. I told you we are interested in this continuum between tolerated splicing noise and disease causing splicing mistakes. And now for this, we went to another database because the GNOMAD is the general human population. It's not disease specific, although it contains some diseased sequences. And now what we did was to compare two data sets, whole genome sequencing for a number of genes that are known to be involved in cardiac disease. And we compared splice variants present in over 800 patients and approximately the same number of non-diseased adult individuals from the GITEX database. And when we compare the proportion of splice variants for the same gene in the two groups, they tend to be very similar. So both the patients and the normal individuals, they have more or less the same number of splice disrupting variants in their genes, indicating that this variance is tolerated, it's not a problem, except our gene of interest, the myosin binding protein, where the majority of splice variants are present in patients, and very few are present in normal individuals. And when we do a statistical analysis, this gene is very significantly enriched in splicing variants. So this gene is particularly sensitive to splicing perturbations. So this is telling us that not all genes are equal in terms of tolerating variability. So, although gene level context matters, depends on some genes are more tolerant than others. In general, why are so many splice variants tolerated? And so we decided now to quantify the number of missplicing in these tolerated, so non-disease causing splicing variants. And this is one of many examples. And what we observe is that when Splice AI predicts an abnormal splicing, it tends to be there. But the number of transcripts that have this variable splicing is extremely low. So this is data from RNA-seq, and only 1 to 2% of all transcripts contain this pseudoexon that is normally not present. And then when we do QRT-PCR quantification, and remember I told you that many of the misspliced transcripts, they are degraded by nonsense-mediated decay. We can inhibit nonsense-mediated decay to give an idea of all the misspliced transcripts. That's what we did here. We use a translational inhibitor to block NMD, and you can see that the qPCR validated about 1% like the RNA-seq, but when we inhibit NMD, the number of these misspliced transcripts increases as one expects, because these transcripts are normally degraded. So even with the degradation, up to 3 transcripts had this missplicing, which indicates that the intrinsic error rate of splice site recognition is very low. There is a strong tendency for the spliceosome to have a high fidelity, to do the proper splicing and avoid missplicing, even when there are genetic variants that would promote missplicing. So this brings a number of questions. If I showed you that in a disease-caused situation, the number of missplicing was 20–30%. In a tolerated missplicing, the numbers is up to 3%. So there is this discrepancy in amount of products of splicing variability. Is there a threshold that distinguishes whether splicing variability is tolerated or is deleterious? And what controls the accuracy of the spliceosome? It seems that the spliceosome is extremely accurate, but are there extrinsic or environmental factors that change the accuracy of the spliceosome? In particular, does aging or stress increase splicing noise? Because there is recent evidence indicating that with age, the splicing variability increases. I used to think that this is bad, so we are aging and our cells are now making more mistakes. But again, as I was listening to Francesca, maybe we should think about this as an attempt to adapt to this stressful condition of aging. Is fidelity decreasing to try to increase variability and try to adapt to this new environment, which is the aged cell? I just talked about it some minutes ago. And so what we are now pursuing in our lab, because we are an experimentalist lab and we would like to understand the mechanisms of RNA biology processes, we are trying to address this question: what controls mechanistically the accuracy of the spliceosome? And we thought that we could devise screening systems to identify factors that are required for spliceosome fidelity. This is just one preliminary finding that made us think that we might be in the right track. Obviously, we are thinking about setting up genetic screens, but we started by a pharmacological screen. There are a number of drugs that inhibit different components of the spliceosome and spliceosome-associated proteins, and we have been testing them, and we came up with one of these drugs called indisulam that acts by promoting the degradation of RBM39. RBM39 is an RNA binding protein that is known to be a co-activator of the spliceosome. And when we treat ourselves with this drug and we confirm that RBM39 decreases, is degraded, what we see is a significant increase in the proportion of aberrant transcripts. So indicating that this particular protein is important for fidelity, and in the absence, now the spliceosome makes more mistakes or is more variable in its decisions. So with this, I come to my conclusions. So I've shown you that although individually rare intronic splicing disruptive variants are widely distributed across the human population, despite the presence of these variants that promote splicing variability, the spliceosome tends to retain overall accuracy, so that only a small proportion of misspliced transcripts are produced. And we think that this is the main reason why they are tolerated. Then some genes, as the C protein, but for sure there are others, are particularly dosage sensitive, so meaning that a certain reduction in the total amount of normal transcripts is sufficient to cause a phenotype leading to disease. And finally, my proposal that understanding the mechanism that govern spliceosome fidelity may reveal why splicing accuracy declines with age or under stress. And these are factors that shape disease sociability and progression. So we are very excited with the idea of pursuing these mechanistic type of projects. And with this, I thank all the members of my lab, all our collaborators, our funding agencies, and you for your attention. Thank you very much.